looking for. Thank you, ma'am. Yeah, I was pleased that people were willing to let me know that since they were going to miss the meeting. I'm going to have to come up with some kind of stand-up routine, because yesterday we had a meeting too, and we had to wait and nobody was talking, and I felt awkward, but I'll come up with something next time.
So Kelly, are we waiting for more people?
I'm sorry. Yeah, we have a total of 8 who had signed up. I'm sorry, 11. The other meeting has 8. Y'all were the top meeting with the top responses. Yay. If you want to start now and they can catch up, or they can watch the recording, that's cool with me. I have us booked until 2:45. So if we really get into a discussion, y'all can knock yourselves out till 2:45. So you want to go ahead and get started? Dr. Kwan?
Yeah, I think that sounds good.
Okay, I'll bow out. Thank you. Holler if you need anything. Thank you.
Okay. All right, welcome everybody. I'm Esther Kwan, and we have with us Professor Sheikh. And so I'll just go over briefly what the course entails, so you just have a idea what it's about. And then we'll be happy to take any questions afterwards. But I just have like 5 minutes of presentation. All right, everyone can see that? Oops. All right. So this is for winter quarter of BENG 277, so Tissue Engineering Laboratory. I'm the primary instructor, and we're lucky to have Professor Sheikh with us as well, who will be teaching a module. And we have a teaching assistant, Shivani Shukla. The class is Tuesday, Thursdays, 12 to 1:50 p.m. Generally, it will run the full time, although there are some classes that will end a little early. And we'll be in Powell Folk Bioengineering Hall, room 108. So it's on the ground floor there. It's right across the bioengineering office. So the course is designed to give students a hands-on experience with experimental techniques in tissue engineering. And so we'll go through a variety of kind of basic and well-known types of procedures and questions throughout the course. There'll be 5 modules, and each module is roughly 2 weeks, and each module will be done in groups of 2 students generally. And the ways that you'll be evaluated is your preparation and performance. So this is kind of like attendance, and you're using your lab time appropriately. 60% of your grade will be lab reports, and then 30% of your grade will be a final paper, which will be based on an independent project that you'll do in the course. Here is preliminary schedule that's set so far. So there'll be 5 modules. Each will run roughly 2 weeks. And at the end of the course, there will be an independent project, which is basically another module that you'll be working on for your final paper. So the 4 planned modules, you will be provided a framework. So you will have an objective. So what are you trying to study, and a reagent list. But the details of specifically what you'll be doing will be generated by you and your teammate. For example, well, I'll give you some examples as we go through each of the modules. So you are expected to read some of the literature and prepare protocols ahead of time and be ready for the course or the class time. Of course, an instructor will be there as well as the TA to help you iron out some kinks, or if you have questions prior to the laboratory start time, to help you navigate that. So let's just go through each of the modules. The first module will be decelerized tissue. And so here we'll be using a piece of pork loin from the grocery store and decellularizing it using some standard techniques. So in this module, you'll learn how to decelerize tissue. And so the reagent list will give you a hint on what kind of decellularization you can look for. In this case, we're using sodium dodecyl sulfide, a detergent. You'll do histology staining in class, and we'll provide the specific instructions for that, and then perform light microscopy on the histology that you do. Another module will be the synthesis of natural hydrogels. And so one will be a collagen hydrogel, which you'll do scanning electron microscopy to image fiber diameter. And then a second gel will be sodium alginate gels. And you'll be testing those mechanically using an ELF machine, which is basically just looking at the viscoelastic properties. So these are two very commonly used materials to form gels. So we'll be guiding you towards those. And then another module will be synthetic hydrogel. So instead of using naturally occurring polymers, this one will be using polyacrylamide to create different stiffness of gels, and then studying stiffness dependent behavior. And so here you'll be synthesizing polymeric gels, culturing cells on them, staining cells on them, and then imaging them with fluorescence. Okay, and then the last module, Dr. Sheikh, I did create this little outline slide. Feel free to use it or ignore it, but just to key you up.
Yeah, so my module is really related to the use of neonatal cardiomyocytes as a model system to study hypertrophic growth, because that's often deleterious when it comes to heart failure when you get too much hypertrophy. And so we'll use this as a platform to better understand what types of drugs may sort of intervene or make things worse. And so we'll be using a lot of, so the cells are already differentiated, but we will add various molecules in order to push these cells to become more hypertrophic. And you'll do some experiments into what types of drugs you'll use. You'll have your own combinations and have hypotheses that you'll be testing with this. And then we will use immunofluorescence microscopy as a method to also measure some of these hypertrophic signals. And you'll go through what types of markers and assays you'll do within the module itself.
Okay, so that was all that we had prepared, just to give you an orientation for what to expect for the course. We'll open up to questions. I would comment that I'm going to set enrollment soon. There's only a limited spots, so there's 12 spots. So anyone here who wants to join, you have, there is a spot for you and you have priority. But after that period, I'll start letting in the general admission to non-interfaces students. So please send me an email if you're interested in joining, just to make sure I save a spot for you. And you can also copy Kelly, who I think also has a list for the enrollment. But from here, I think happy to take any questions about the course.
I guess I can ask a question. So my background is not in engineering. I'm in the biomedical sciences program. And so I was kind of wondering if there's any like background knowledge that you're expecting us to have. I've never done a lot of these like gel based experiments or like even really read about them. So that would be totally new to me. So I'm guessing like what kind of knowledge do we have coming into this course?
Yeah, that's a great question. So this course was designed for people who either don't have much engineering background or people who don't have biological background. So if you have one of the two, you're going to fare quite well. And so no expected knowledge no expected background is required to do well in the course. You will need to be able to handle reading some primary literature. That's where you'll be basing your protocols on. So that would be really the only expectation.
And at least for my module, you know, I give a list of materials to read. So you'll have a concise list. You don't necessarily have to go on PubMed and go crazy or anything.
I guess another question is like, what is tissue engineering? Like, what does it mean to engineer tissue? That sounds like a basic question, but I guess kind of like, what's the overall like theme that connects all of this?
That's a great question. So the triad of tissue engineering are that the basic components of something that you would tissue engineer are cells. So where do you get your cells? They could be primary cells, cell lines, they could be autologous, they could be allogenic or xenografts. The second part of the triad is the matrix. And so you'll see that a lot of the course design is on studying or components of the matrix. And so that can be natural matrices or synthetic matrices, and then controlling their structure and their topography and all these sorts of things. And then the third part of the triad is soluble factors. And so these can include like stimulus for growth, like growth factors, or other things that you'd add to, you know, support the growth of your material. And the overall goal is to recreate and recapitulate some sort of biological behavior of an ensemble of cells in a organized structure. And so I think Dr. Shaik's module has components of the cells and the soluble factors. The decelerized component has like the ECM component. The synthetic and natural hydrogels modules, they have components of the extracellular matrix, and then some of them have the cell component as well. But there is a course 241A, which Introduction to Tissue Engineering for graduate students. So if you're interested in learning more, that's also a good course to take. I think Dr. Chrisman teaches that course.
And I, you know, I work very collaboratively with bioengineers, although I have a very molecular cell biology focus. And so oftentimes, you know, it's difficult to crosstalk with the other side unless you have a particular question. So I think, you know, this course also very nicely attunes you to what types of questions, maybe even in your own research, that you can think of. And, you know, it makes you think out of the box instead of, you know, whether it's from a biological or a engineering standpoint. An example of this is I also work with Karen Chrisman, and we often, you know, want to mature our cells because a lot of cells in a dish sometimes don't have the right cues. And so, you know, an ECM from a naturally derived, you know, the pig heart, for instance, from a particular tissue, like the organ of choice that you want those cues from, could give you that particular, you know, I guess the factors or the cues to sort of direct those cells to be more mature. So this is some examples of crosstalk. And I think not enough is out there on people that are looking from both sides. So I think this is like one of the coolest courses in terms of that, because, you know, you can have either background, but you have to have some basic knowledge, I think, that can give you that insight into, you know, putting it towards your own questions in your own research.
Thank you. That's a really helpful perspective. I guess I can ask one more question. What do you think the time commitment is going to be outside of the lab hours?
I think you can expect a little bit depends on how much experience you have with each of the modules, but it is designed to be able to complete it with within 10 hours a week. So you'll be provided the skeleton of each experiment and some background reading. And you can do, of course, as much reading outside of that as you would like. So, so each module, you'll read a handful of papers, 3 or 4, and then read the experiment outline and do some planning. The independent project might take a little bit more work, although you kind of know it'll be coming up at the end of the quarter, so you can prepare for that ahead of time. Yeah, and then in terms of the actual experimental time, it'll be strictly the 4 hours a week. There's a question also from Kristen Garcia. Is it okay if no wet lab experience, if you don't have any wet lab experience? And I would say, yes, it's okay. So we'll be, there'll be a TA to help and guide. And so I think you just need to let us know before the, the period starts that, you had some questions, and we'll be happy to help you with that throughout the period. So it is a completely introductory class.
Yeah, I know in my module, I kind of talk about aesthetic techniques, and how to treat cells. I mean, especially primary cells are, can have the ability to contaminate, but we do use antibiotics in order to prevent that. And since you're not really culturing them for that long, it's okay. So they are kind of robust in terms of, if you're a little bit harsh with them, so they can withstand some stress. But at least within my module, which is more wet lab related, we really guide you, and everything is done in class. Even the microscopy work to analyze the stuff is done in concert with, my people. So it's kind of, you're really guided through everything. So it's not like you're left alone and you have to double check your protocols with us and all of that. So we know you're on the right page and track and things like that.
Yeah, and I also comment that you'll have a lab partner, so that between the two people, you can fill in gaps if you have if you don't have knowledge in one area, your partner might be able to help you out. All right, any other questions? Feel free to type them out if you are unable to ask them out loud.
Well, we hope to look forward to seeing you guys in the class.
Yes.
In person.
Yes, in person. Yeah, so just to reiterate, please do send me an email if you're interested in enrolling, just so I can make sure to save your spot for you.
Thank you so much, Dr. Kwan and Dr. Sheikh. I appreciate that. And thank you, students, for attending today. I will give you information about the recording as soon as possible, as soon as I figure out how to get it down from the cloud, hopefully today. And as always, email me at KJ if you have any questions. And other than that, have a great day. Thanks, everybody.
Thanks, Kelly.
Bye-bye.
Thanks. Bye.
Take care.
You too. Bye-bye.